pleasure to be here, and I've really been enjoying the fantastic array of talks that I've been listening to. So today I would like to talk to you about RNA chemistry and how that might have played a role in the transition from the abiotic to the biotic world. So we're all here for one reason or the other, but it has something to do with how you go from the physics of making stars, planetary systems, galaxies, to this, which is metabolism, which keeps all of us alive. And there's a huge divide between the formation of planets and how metabolism emerged. And really one of the nice unifying principles behind the RNA world model is that it gives key insight into how that might have happened. And so a lot of my research has been dedicated towards figuring out if RNA can actually do the type of chemistry that modern cells with proteins in them seem to do as a matter of course. All right, so if we were to think about the transition from the prebiotic to the biotic world, we might, I think, all agree that a scheme somewhat like this, where we don't need to pay too much attention to the straightness of these lines, could have taken place, where you have a lot of very simple abiotic molecules which have a very high abundance on a planet. And through some prebiotic chemistry, you get reactions that build up increasingly complex molecules that ultimately can go no further before you have life. And the nice feature of the RNA world is that if this abiotic chemistry could get to the point where you can build polymers, nucleotides, which are the substrates for making RNA, then you could imagine how you can go from this prebiotic situation to a biotic one, which would be down here. So how might that actually look in practice, plotting on a graph like this? So if you actually manage to get to a point where you can get RNA molecules or polymers of a big enough size that they can do chemistry and can replicate themselves, then this blue component of this graph might emerge from this abiotic chemistry, where you'd have large polymers that are able to do chemistry and actually replicate themselves in a way that could not have happened before they emerged. And I think integral to that, and we've heard a lot of talks this morning on this subject, is that you might need some sort of partitioning between the outside world and the inner world where these polymers are replicating, where you would have some perhaps very porous initially, but you would have a membrane system that would keep these replicating polymers away from the outside world. So as evolution proceeds, now that you have a way to evolve, because you have an information system that is in a compartment, you need more and more complexity. You need to get a metabolism going that is not completely dependent on the abiotic chemical processes that were initially there to generate life. And as you do that, you need more information, and you need the ability to convert that information into catalytic power that is able to make the small molecules that perhaps originally you depended upon from the environment, but that are no longer around because you're now starting to compete for them, and there presumably weren't that many around to start with. So you need some way of making those molecules from simpler compounds. So for example, in the RNA world, these polymers are RNA strands, and nucleotides, presumably if you could make them, and when I first came into this field, I was very worried that it wouldn't be possible to make nucleotides prebiotically, but nucleotides, from the wonderful work of Pauner and Sutherland, may well have been possible in the abiotic situation. Even though they're synthesized abiotically, I don't think they would have been very abundant on the early Earth. And these living systems would have consumed them very rapidly. So you would need some mechanism inside this living system to make the nucleotides that are keeping the polymer-based system going. And as this proceeds, you eventually rely on simpler and simpler molecules, and life can take over larger and larger portions of the Earth or other planets' surfaces. All right, and eventually you get to the modern day situation where the permeability of a membrane can be quite small, and you have very elaborate mechanisms to actually import the food that you need to survive and keep your information separate from the abiotic world. All right, so with that kind of transition in mind, I focused on, in my laboratory, figuring out how easily RNA can manipulate small molecules smaller than the monomers that actually make up the actual nucleotide chain of RNA itself. And if it can do that easily, that's a good sign. If it can't do it so well, then this whole idea of an RNA-dependent metabolism is perhaps more suspect. So I'll talk to you briefly about nucleotide synthesis in RNA. So what we've done is selected, using the powerful tool of in vitro selection, a range of RNA catalysts or RNA ribozymes that are able to promote on the ends of themselves a nucleotide from two simpler components. And the building blocks that we chose to use are PRPP, an activated form of ribose that we use in all of our cells today to make nucleotides, and a free base, either a pyrimidine or a purine, which when you incubate with these activated ribozymes containing the activated ribose or PRPP, are able to make a glycosidic bond and a nucleotide, an important building block of RNA. And just to show you what these look like, here are two secondary structures of two different types of pyrimidine nucleotide synthases that each are able to make a nucleotide at their terminus. All right, so that's a powerful fact that you can make nucleotides with RNA. What's interesting, I think, about how this is achieved is that it mimics very closely the kinds of specificities that you see with protein enzymes. So if you test how well these small, the pyrimidine nucleotide synthases are able to recognize a substrate that they're actually reacting with, this pyrimidine, and you make alterations to the pyrimidine and ask how well they react, you see very good specificity. You see drops in activity by many orders of magnitude, at least 3 orders of magnitude in most cases for the pyrimidine nucleotide synthases. If you look at the purine nucleotide synthases, if you substitute positions on the purine ring, you see, again, dramatic losses in activity, which is very reassuring because that would tell you that if you have other molecules, if you need to do chemistry with different types of small molecules, RNA can actually do this. You see at a number of positions that you have very specific, that the chemistry is very dependent on having the right molecule there, except for this amine right here. And if you look at natural protein enzymes that are reacting or building nucleotides with guanosine, it's exactly this amine that is not recognized by the protein enzymes as well as these RNA enzymes, implying that both RNA and protein are, we know for sure protein is able to fundamentally discriminate between small molecules, but the way that they're recognized them may have some commonalities. All right. Here's another very interesting feature of RNA, and it's chemical promiscuity. It's perhaps at the other end of the specificity that I was just talking about. I think you need specificity in a metabolism. You also need to be able to evolve very rapidly other types of chemistry. And so a student in my laboratory asked the question, what would happen if you reacted just an unactivated ribose sugar with a purine base? What kind of products would you get? And these presumably would not be nucleotides, but there would be some other product that might have some relevance to metabolism. So what he found, quite interestingly, was when you do this selection, the aldehyde at the end of this ribose, when it's in the open form, can react with the guanine base that it's incubated with and form a product. And when he tried to, when he did a control experiment to see how preventing this circularization and open form, by disrupting this equilibrium between open form sugar and circularized sugar, by actually putting PRPP on the end, which stays in the closed form, he found indeed that this a particular aldehyde-dependent chemistry was inhibited, but that a new product, a much more rapidly forming product, was formed. And not to dwell on this graph any longer, but to show you what's going on for the first chemistry, where you just have the aldehyde, we think that a Maillard-type reaction is going on. This is familiar to all of you if you take onions and and meat and cook it in a frying pan, and the nice smells that you get. I mean, this is a very simple aldehyde-dependent chemistry that's going on, and that he had selected that with this ribozyme to do a chemistry that, well, frankly, is perhaps not that exciting, but does couple a base to a sugar in a novel way. The intriguing part, though, is that when you put the PRPP on the end, and I don't have time to show you the characterization here, is that you get a completely different chemistry, which is nucleotide synthesis. And so if you had ribozymes early in the evolution of life that were doing one chemical reaction with perhaps a sugar, there's no particular reason to think that it would have been at all complicated to make a new form of chemistry emerge almost by fiat. I mean, there would have been no change in the sequence of this RNA required to go from those two different chemical reactions. All right. So the final subject I'd like to talk about is nucleotide polymerization. And there's been some very exciting progress in this area, and I just really like to finish with a cautionary tale on this particular subject, which is that we have a long way to go before we can make a replicase in the lab. So I'll just show you the exciting parts first. So this is the class 1 Bartel and Shostak ligase. It very efficiently joins two pieces of RNA together, and it has been engineered over a decade or more into a ribozyme that's able to polymerize an RNA primer that's hybridized to a template. If you work in my lab by Hani Zahar in 2007, did a selection where we were able to enhance the ability of this polymerase to extend a template from these two panels on the left and on the right are showing extension of these two primer template sequences. And you can clearly see for the longer primer template, this particular selection took the old variant selected in Dave Bartel's lab when I was a postdoc there, took this old extension profile to an extension profile of over 20 nucleotides. Now, recent work has shown that you can do much better than that, and I'll show you the most recent work on the next slide. But what we have found in my laboratory recently is that tethering of the primer template to that particular ribozyme polymerase can dramatically enhance polymerization efficiency. So if you just look at the original polymerase for both tethered and untethered substrate, you can see that if you tether it at a range of different sites on the polymerase, you can actually choose sites where polymerization can go up by factors of 20 to 30 over the untethered polymerization, which is shown down here as a control. So there's specific spots on the polymerase, particularly its 5' end, and in one of the loop structures of the polymerase, where you can tether a primer template and get very dramatic increased rate of polymerization. And that's entirely consistent with this beautiful gel from Holliger's group that was recently published, where they did an in vitro selection. They find a couple of mutations with respect to the ribozyme I just showed you, but I think most critically, they're tethering the primer template to the polymerase, and that they are using a repetitive sequence that is very good at being extended, and you get this beautiful extension out to at least 95 nucleotides. Now, this is a very impressive result, but there's a number of things that we're going to have to work on really hard in order to make something that can copy a piece of RNA quantitatively. As Jerry Joyce pointed out this morning, this amount of extension up here is about 1 part in 10 to the 3 or so. And we'd like to make that much closer to 1 than just a small fraction. And so there's a number of very critical hurdles that we have to pass before we'll be able to turn this RNA system into something that's able to copy routinely and reliably RNA into its copy. So just not to stress this in any detail, one of the surprising things is that the accessory domain, this new domain that was attached to the ligase core, this is the ligase core from David Bartel in blue, and the accessory domain is the functionality that has been appended to it that allows the ligase core to do polymerization. The most surprising thing I think we found recently is that this doesn't actually really need to be, it exists as a separate domain from the ligase core and is folded by itself, and in trans, just adding these two in a tube together and a primer template, you get extension. All right? So if you tether this accessory domain at a number of sites on the ligase core. Surprisingly to me, this accessory domain could confer the ability of polymerization in a number of different sites, with several sites being the best, in particular these two constructs here, either at the 3' end or at the 5' end. But quite surprisingly, this accessory domain seems to know how to dock down onto the ligase core so that it can construct a polymerase ribozyme. And if we look at the minimal structure of this, which is built out of a total of perhaps 150 nucleotides, depending on how you want to create the linker here, we have a ligase core, which can dock the primer template, and we have this minimal accessory domain that is responsible for ways that we still imperfectly understand, for positioning nucleotide triphosphates for the extension of the primer template, which is docked to this complex. One of the things that's really noticeable or notable about the whole system is that it really does not like to hold on to the primer template. Tethering has been a really important aspect of getting efficient extension. And just to show you how this might dock, it's pretty clear that at least one of the tertiary interactions involves these two tri-loops on the ligase core and the accessory domain is essential to correctly position these two structures together that really, as I said before, exist as two separate modules for this machine that does nucleotide incorporation. So how do we proceed from here? How do we make this into a better machine? How do we increase its processivity? I think these are really key steps that have not happened yet, but some ideas. It should prove possible, based on our understanding now of how the ligase core and the accessory domain relate to each other in three-dimensional space, to select another domain that makes it much more processive. And so if we could make this whole module hold on to an RNA duplex or a single-stranded RNA, perhaps might be harder, but equally could be tried, we could make a complex that could perhaps much more processively fill in a primer template so that we could copy this black strand. If we could do that, we might be able to solve another key problem with this RNA polymerase ribozyme, which would be how do we actually avoid a dead-end state? So if you fill in completely this black template with this black primer, you end up with a duplex. And that is not very useful for replication unless you have a polymerase that can actually invade that duplex so that it can make a fresh copy. So if we could increase the processivity, perhaps with this new domain yet to be selected, there is a chance that we'll be able to do strand displacement, as I'm trying to show you here, where if you hold on tightly enough without recognizing the duplex that is emerging from it, you should, we hope, be able to displace this red top strand and make something that can truly replicate both itself, which would arguably be something that could be used used in the test tube to simulate an early replicase in the RNA world, but also copy other ribozymes that have metabolic properties that I tried to show you earlier have important consequence in the RNA world. So with that, thank you very much. And I'd just like to acknowledge these people who did the work.